Hello dear students. Yesterday I just stopped here while explaining the formation of vesicle, that is coated vesicles. So here the coated vesicles are going to get formed by the clathrin. So this particular clathrin is a, you can see here, this is the structure of clathrin. It's called triskelion because it will show three legs. It will be made up of three heavy chains and three light chains. So heavy chains are going to have N-terminal larger domain, like a ball-like domain, with which they are going to interact with the other clathrins as well as the adapters and forms a polymers. And the light chains are going to be involved in association or a structural stabilization of this clathrin triskelion. And the C terminal is going to be joined. Each of the heavy chain C terminal will be joined together to form this triskelion structure. So it will have this distal segment anchor linker like that, so which are all going to play an important role in polymerization and giving out a cage like structure, which you can see here. So they can form hexagonal cage or soccer ball type cage. Okay, so like different type of cages are going to get formed by these particular clathrin triskelion. And clathrin, whenever it is going to involve the formation of vesicle, it is going to form vesicle with the help of an adapter protein. So this is the structure of an adapter, particularly it's called as AP2 adapter. So these adapters are going to have this alpha, beta 2, mu 2, delta 2 type of sigma 2 type of subunits. So this particular structure which you are seeing here, this is AP2, wherein this is going to involve in the formation of vesicles at plasma membrane during endocytosis. So you can see that the mu subunit, what this mu subunit is going to do is it's going to interact with the endocytic motif. Endocytic motif means the receptors which are going to be present in the membrane, which actually interacts with the ligand, or the proteins or the cargo which is going to be internalized. So these particular receptors on their cytosolic side, they are going to carry a sequence of nucleotide sequence of amino acids which are exactly recognized by these AP2 and bind with. So that sequence you can see there, that is YXX5, where Y will be a tyrosine and XX can be any other amino acids, and 5 is going to be usually an hydrophobic amino acid. On the other hand, we can see the beta and alpha is going to have the appendages. So these appendages are going to involve in binding with the clathrin. Okay, so thereby they are going to establish a linkage with the clathrin as well as the receptor. So you can see there the alpha is going to have a repetitive sequence or a motif of DFF or DFW. So this DFF or DFW is nothing but aspartic acid, phenylalanine and tryptophan. So this particular motif again plays important role in linking the heavy chains, the N terminal of the heavy chains of clathrin with these adapters. Okay, so these adapters and the clathrin together, they facilitates this formation of vesicle. So how it is going to get formed here is, so I was talking about the priming process which involves the ARF factor. This ARF is a protein that is the long form of ARF is going to be ADP ribosylation factor. This ADP ribosylation factor, it is going to be an important protein molecule which partially you can say it decides where exactly the formation of vesicle is required. So this particular protein belongs to the RAS superfamily of protein molecule, so which includes a large number of proteins carrying the motifs like RAS. So this particular protein is going to get activated by the exchange of GDP with the GTP, and this particular exchange is brought about by the protein which is a GEF or guanine nucleotide exchange factor called as ARNO, that is ARF nucleotide binding site opener, we call them as. So this ARNO, what it does is it's going to help in the exchange of this particular guanine nucleotide, that is ADP with the ATP and then activates the ARF. This activated ARF with it, sorry, yesterday I was telling it's palmityl, it's myristyl group, which is a hydrophobic chain through which it's going to grow itself into the plasma membrane, where exactly the formation of vesicle is going to be required. So what is going to happen here is, through this ARF is going to interact with the membrane and gets hooked up into the membrane. Once ARF gets hooked up into the membrane, then it is going to start recruiting other subunits like AP2. So AP2 or an adapted protein, I can say AP2 or AP1. Okay. So when this adapted protein is recruited, so this particular adapted proteins, now it starts interacting with the receptors carrying the signal sequences. So I told you some of these particular receptors is going to have YXX5 as a sequence within the cytosolic side, or even it can have D or E XXXLL sequence also. So once it finds this particular exposed YXX5 or this particular DRE XXLL sequence, so then it is going to interact with that particular receptor and starts binding with that particular receptor. Means a firm contact is going to be made between AP2 and that particular receptor cytosolic domain. So then what will happen is that this interaction of AP2 is going to start recruiting the clathrin triskelion. So the AP2 interacts with the clathrin triskelion through its appendages and forms a, means it initiates the polymerization of clathrin around that particular part of the membrane. This polymerization of clathrin starts pulling that particular membrane inside it, just like a suctional force. Thereby thereby it is going to form an assembly thing. So once the assembly is almost getting over, that is due to the pinching off of this particular membrane forcibly by the polymerization of the clathrin, so what will happen is that it leads to the formation of a small neck-like structure, so wherein the protein called dynamine will come into play. So it is a 870 amino acid residue having a GTPase protein. So what this dynamine is going to do is, this particular dynamine goes and starts polymerizing at the neck of this particular pinching means at the neck of this particular membrane. So the this particular dynamine, it forms an helical oligomer which wraps around the budding membrane and squeezes this particular region down to a very thin tube. So once it starts oligomerizing and squeezing this particular tube-like structure, so what will happen is that the dynamine, in other words, it's going to pinches off this particular membrane. So once the membrane portion get pinched up and even the polymerization of the clathrin continues, it's going to form a caged or what I can say a coated vesicle. So this particular part is going to be called as a release. So where a coated vesicle is going to get released from this particular portion, and this dynamine, as it is a GTP binding protein molecule and has a GTPase activity also. So the oligomerization of dynamine, in other words, it also causes initiation of the gap, that is the gap domain of this particular protein, that is GTPase activating domain. So there by this particular protein starts hydrolyzing the GTP which is attached to it and converts into a GDP, which makes inactivation of this particular protein. On the other hand, the other chaperonins like HSC70 will come into play. There are number of homologs of HSC70 in humans. We call them as HSPs. So these homologous proteins, what they do is they are going to attach to this clathrin coated vesicle, and they start what you call it as unfolding, or otherwise I can say depolymerizing this clathrin triskelion, means starts removing the clathrin by using ATP. So once and thereby they are going to uncoat the vesicle and releases the uncoated free vesicle, so which now is available for the what I can say for the transportation as well as for the process of fusion. Okay, so this is how the formation of vesicle is going to takes place, and thus formed vesicles are going to be used either as a transport vesicle or as an endosomes. Okay, or even they are going to be involved in the sorting, means transportation of proteins are put to the specific targets. Okay, so this is how the vesicles are going to get formed by uh, the Catherine coated vesicles are going to get formed by the coating process. So the other kind of vesicle formation happens with the COP1 and COP2. So there also we come across with the similar kind of steps as priming, assembly, release and uncoating. So here only the difference is that in case of COP1, as we use clathrin as an initiator GTP protein, GTPase protein, similarly here ARF is also going to be used. In case of COP2, we are going to use SAR1, which is going to act as a GTPase protein. So it's a monomeric protein, belongs to the again same RAS superfamily of protein molecule. So switch the proteins that cycle between the GDP and GTP bound forms. So here you can see the priming process wherein SAR1 is going to go and gets activated by the exchange of GDP, and activated GTP bound SAR1 binds and interacts with the sec12. Okay, that is the one which is actually activates the SAR1, and SAR1 through its N terminus it's going to hook up into the membrane. And once SAR1 hooks up into the membrane, followed by there is going to be coat assembly, which is going to happen with the Sec23-24 complex, which acts as a uh, clathrin protein, and then so it starts assembling, and there will be another set of coating complexes which are also going to be involved in which completes the coating. They are Sec 13 and 31 complex, and then finally that is going to result in the polymerization of the cotomer, and ultimately leads to the release of that particular portion of the membrane as an coated vesicle with the COP2. And once the coated vesicle is going to get formed, later uncoating will happen. This uncoating is going to happen as Sec23 coat subunits. They are going to promote the hydrolysis of GTP, which is there in the SAR1. There SAR1 is going to become GDP, and it comes out of the membrane, and that also leads to the removal of the cotomers, and finally uncoating of the vesicle is going to takes place, and results in the formation of an uncoated vesicle or a transport vesicle, and which is going to be ready to fuse with the particular target membrane. Whereas in case of COP1, similar processes are going to take place that involves in the process, because in the COP1 it initiated by the ARF, and followed by that the subunits of COP1 is going to be involved in the polymerization, and again similarly the uncoating is going to be also facilitated. So you can see there are a number of uh, sequences of amino acids which are going to be present on the polypeptide chain, which specifies their target, and which is going to be read by the adapter protein molecule or even by the receptors, and thereby they are going to involve the formation of vesicles either through COP1 or with the COP2, or even with the clathrin AP2 or AP1 complexes. Okay, so you guys need to remember these particular sequences, which are called as signal sequences, very important for sorting of the proteins into specific destinations through the vesicles. Either if they can be the membrane protein molecules, or even sometimes they can be a free proteins carrying the sequences which are going to get vesicularized and transported in the form of vesicles in the process of secretion, or in the process of transporting a vesicle, means protein, into a particular membrane organelle. So this is how the formation of vesicle is going to happen with clathrin and as well as COP1 and COP2. So here I am trying to concise and reduce the topic as much as possible for you people. So but I suggest you to read these things in very much great detail, because these are little more interesting, and as well as they have a lot of importance in cell biology. So from this particular part of the syllabus, you will be getting an average number of questions, okay, including 2 marks or even 4 3 marks, all are going to be coming from here. Even 5 mark questions are going to come from this particular end. But more than what I can say, the exam part, these are going to be very very important in case if you are going to be attempting any kind of uh, entrance exams and all. So I'll stop here again. In the next coming classes, next coming videos, I'll be explaining how these formed vesicles are going to be involved in fusion, and thereby they are going to help in the mobilization of particular targets into a specific destinations. Okay, thank you, and all the best. So hopefully you people are actually going through the videos, but I just suggest you please go through it and try understanding it. In case if you find difficulties, please do contact. At the end of these particular videos, I'll be giving you the list of references, so even the topic wise references also I'll tell you, so which may help you to understand and study well. Bye.